Good morning, dear students. By the last video, we have seen the early embryonic development in C. elegans, and today we are going to start the early development in Drosophila. What is the peculiarity of development in Drosophila? In the case of Drosophila, until the 13th nuclear division, there will be no cellular membrane formation. We can see that a nuclear division here, and until that 13th nuclear division there is no cellular membrane formation. So that entire nuclei share a common cytoplasm, and that cytoplasm it diffuse throughout that multinucleated cell. Okay, and the dorsal ventral axis as well as that anterior posterior axis. According to that axis, the specification of cell that occurs because of the interaction of materials that present in the cytoplasm. How that axis is forming? Because of the interaction of materials present in the cytoplasm. Okay, and one more thing, in this Drosophila, the initiation of axis formation, it is controlled by the position of the egg within the mother's ovary. That's also a special thing. While we talk about ascidians or nematodes, in those cases, that axis formation is controlled by the point of entry of sperm, and that determines the axis formation in the case of ascidians and nematodes. But in the case of Drosophila, that axis formation initiation is controlled by the position of the egg within the mother's ovary. Okay. Next is about the fertilization in Drosophila. In the case of Drosophila, the activation of egg is happening immediately after ovulation. What is ovulation? Ovulation is the process of release of mature ovum from the ovary. But in the case of some higher forms as well as some other invertebrates, that activation of the egg is happening only after the entry of sperm. Then what is this egg activation? Egg activation means the egg that is stalled at the meiotic metaphase 2. It is resuming the next stages in the meiotic division. So that is activation. In the case of Drosophila, that activation is happening just after the ovulation, before fertilization. Okay, that is one difference. And regarding the axis specification, that also starts before the entry of sperm into the egg. And through which region the sperm enters the egg in the case of Drosophila? Through a structure called a micropyle. It is a tunnel-like structure that can see in the anterior side and also dorsally, somewhat dorsally positioned. And only through this structure, a sperm can, Drosophila sperm can enter this egg. And this is the structure. It also helps to prevent polyspermy, because in Drosophila there is no cortical granules to prevent polyspermy. Here the main structure that prevents polyspermy is the micropyle itself. Okay. Next, regarding the sperm of Drosophila. The Drosophila sperm is a longer one. Its tail is too long, and when compared with the size of a human sperm, it is 300 times longer. And the long tail of the sperm of Drosophila prevent other sperm from entering the Drosophila's egg. And one more difference is there when compared with the higher forms. In the case of higher forms, the sperm break down its cytoplasm. Sperm break down its cytoplasm before entering the egg. But here there is no such breaking down of cytoplasm. Sperm as such completely fully is entering inside the Drosophila's egg. Okay. Next we are going to see the cleavage in Drosophila. In the case of most of the insects, the cleavage is superficial. And what is superficial type cleavage? Superficial type cleavage means it is a type of meroblastic cleavage itself. What is meroblastic cleavage? I will explain to you later. In superficial cleavage, even if mitosis is happening, cytokinesis is not taking place. Division of the cell is not taking place. Only the nucleus is keeping on dividing. And what results as a result of such type of division? A cell with a numerous nucleus, a syncytial condition. So that is superficial division, superficial cleavage. What is meroblastic cleavage? Meroblastic cleavage is nothing. It is an incomplete division. That means only a portion of the egg divide. That may be because of something that is hindering that cleavage furrow to completely pass through the egg. Sometimes it may be the enormous amount of yolk present in that egg cell. That may hinder the deepening of the furrow to the entire region of that egg. So such type of cleavage is called meroblastic cleavage. Okay. Here we can see the superficial cleavage in the case of Drosophila. And why this superficial cleavage happens here? One reason is the presence of a bulky yolk that is placed centrally. And after it completes 8 series of such nuclear division, how many nuclei are forming here? About 256 nuclei are forming here. Because of the lack of enough space, I can't show all the nuclei here. And also you need to notice that cell membranes are not forming here for each and every nuclei. And after the 9th division, look here, some 5 nuclei migrate to the posterior pole, and later it develops its own cell membrane, and in future it becomes the gametes of the adult. And then after the 10th division, the remaining nucleus, it moves to the periphery, and undergoes 4 more divisions. And as it finished this much divisions, then we can call this embryo as a syncytial blastode. Syncytial blastode means we know that what is the syncytial condition. That means a cell with the multi-nuclei and without individual cell membrane for each nucleus. So syncytial condition, simply syncytial condition is a multi-nucleated condition. And one more thing, as we look here, we think that all these nuclei are sharing the same cytoplasm, but actually it is not like that. The cytoplasm that's seen in the vicinity of each and every nucleus is distinct from that present that of the other nuclei. That means the protein, cytoplasmic protein that present near to this nucleus is different from that present near to this nucleus. And also those nuclei that moves to the periphery, it gets surrounded by so many microtubules and microfilaments. Okay, and also along with this, we need to note one more term, that is energits. What is energits? Energits means it is the name given to the nucleus along with its cytoplasmic island. Nucleus with its distinguished cytoplasmic island is named as energits. And after 13th division, then that membrane starts to fold. The membrane starts to fold. Starts to fold by forming furrows between the nucleus. And as a result of this furrowing, later uh, cell membrane will form for each nucleus and the embryo become a cellular blastodome. Cellular blastodome. It is mentioned here, become a cellular blastodome. That means each and every nucleus that time gain its own cell membrane. Then how this invagination of this furrows means invagination of this plasma membrane is happening because of the help of microtubules and microfilaments that first come and assemble near to each nucleus. Because of their action, that constriction, that furrowing of this plasma membrane occurs. And as this embryo attained this stage, that is the cellular blastoderm stage, that time that embryo contains how many cells? Around 6,000 cells be present. And when this stage attained after 4 hours of fertilization, then that's about the cleavage in Drosophila. Next is mid-blastula transition. As we see in the former slide, it is the stage of the embryo that have reached the 10th division. And we know that after 10th division, all the nucleus will move to the periphery and it requires to undergo 4 more divisions. And the important thing is that 4 more divisions require more time, that is from the 11th division to the 14th division. So we are going to see the time requirement in each cycles of division of the nucleus. From first division up to the 10th division, it requires 8 minutes each to complete each division. And coming to the cycle 13th division, we know that after 13th division, each nuclei will develop its own membrane and will form a cellular blastoderm. So cycle 13, it is the last phase for the embryo to be in the syncytial blastoderm, and it requires 25 minutes to complete that cycle. Then coming to the cycle 14, this we know that during this cycle or after that cycle, that embryo turns to be a cellular blastoderm. And coming to the nature of division, it is asynchronous. Asynchronous means the division of the time required for the division of each nucleus, each cell is different. Means time requirement is different, it's not same. Some take only 75 minutes to complete the division and some take 175 minutes to complete the division. Okay. Then we are going to understand the mid-blastula transition. What does it means? From the 11th cycle onwards, transcription has started. But by the 14th cycle of division, the speed of the transcription is enhanced, while the rate of nuclear division slows down. Clear? That stage, that condition is called mid-blastula transition. Clear? Next is about gastrulation. When this gastrulation starts, it starts shortly after mid-blastula transition. What is mid-blastula transition? That we already seen in the former slide. And after that, as a result of first movement, then the 3 layers, mesodome, endodome and ectodome, presumptive mesodome, endodome, ectodome, they will segregate or they get separated. Presumptive means what? Presumptive means the supposed to be becoming mesodome layer. So that is presumptive mesodome, endodome, ectodome. So I am just going to show you the position of each type of germ layers. Mesodome we can see in the ventrally in the red color, and the yellow colored region is the endodome, its position. Here also we can see ectodome, and surface portions also there. It is also another form of ectodome, new rectodome. And here we can see a specialized tissue is called aminoserosa. What is the role? It close the dorsal surface of the embryo. I am going to explain you more about this aminoserosa. Aminoserosa, it develops from a small portion of dorsal blastode. And what is its role? It closes the dorsal side of the gastrulating embryo. But as the gastrulation proceeds, it gets replaced by epidermis. And as the aminoserosa get replaced by epidermis, aminoserosa disintegrates, disappears. Then what is the role? The role is that it controls two vital morphogenetic movements of the embryo. Which all those movements? Germ band retraction and the closure of the dorsal side with the epidermis. Then what is germ band retraction, that we will see in the coming portion of this video. Okay. So here I told you that this is the mesoderm. And around 1,000 cells of this mesoderm, it invaginate to form a furrow. These are the cells of the mesoderm. It get invaginated to form a furrow. Its name is ventral furrow. It folds inwardly. That mesoderm folds inwardly to form this ventral furrow. And this ventral furrow later it pinches off from this ventral side and will form a ventral tube inside the embryo. Okay, and after that the endoderm invaginates. Endodome, this is the endodome. We can see that invagination of the endodome. It invaginate both posteriorly and anteriorly. As it invaginate posteriorly, it enclose, it take in these pole cells. These are the pole cells. This one. Then what is this pole cells? Pole cells, I already told you that after the ninth division of the embryo, five nuclei move to the posterior end of the embryo, and later it will develop the cell membrane and will form the germ cells of the adult. I already told you that. So before becoming the germ cells, its name is pole cells. Okay, clear? So that pole cells get internalized by the folding of the endodome. That is the next thing happening here. And next important process that is going to happen is the another furrow is forming here. That is cephalic furrow. Cephalic furrow, how it is forming? The embryo bends. The embryo bends. As it bends, here a furrow is forming, and it demarcates the anterior half, and later it becomes the head portion of the Drosophila. This is cephalic furrow, we can see here. And after that, next, I already told you that mesodermal cells we can see in this portion, and surface ectoderm, that is somewhat seen here. They both converge. Converge means they move towards each other. They converge and then they expand. An expansion we can see here in this next picture. It's very clear. Expand and wrap around the embryo and covers the dorsal portion. And that expansion and that conversion results a ventral germ line or germ band. Its name is germ band. We can see that germ band here, germ band. And what is the peculiarity of germ band? And that germ band includes all the cells that will form the trunk of the embryo. Okay, and we have seen that it expands and wrap around the posterior side of the embryo and covers up to its dorsal side, dorsal surface of the embryo here. And after that, during that expansion time of this germ band, some important morphogenetic processes are happening. Which are those important morphogenetic processes? Organogenesis, organ formation, then segmentation. Segments are forming during this time. Segmentation and segregation, separation of layers, segregation of uh, imaginal disc. It's happening, and also the nervous system is forming during the time when the germ band in its extended form. And the nervous system, as it is formed from the ventral ectodome, that is why in insects like Drosophila, the nervous system is positioned ventrally. Okay, so all these important processes are occurring during the time when the germ band is in extended pattern. And all cells that destined to form the posterior structure, those structures get arranged just behind the head region, here during this expanded stage of the germ band. And after this, the germ band start to retract. It retract from here, and as the retraction proceeds, I already told you that there present another tissue called a aminoserosa, and as the retraction of the germ band proceeds, it will replace the aminoserosa. It reduce the size of aminoserosa, and finally later leads to the closure of the dorsal side. And also after all these process, all the segments that need to be posterior are arranged at the extreme posterior tip of the embryo. Then we can go and see the general body plan of the Drosophila. The general body plan is that body can be divided into head and the tail end, head and tail end. And beyond the head portion, there will present 3 segments. It will become the thorax portion, and after the thorax, again 8 segments be present. That will become the abdominal segments, and each segment is distinct from one another because of the attached structures to it. The first thorax segment, it possesses only legs, first one, and the second one possesses legs plus halters. Halters means such that helping the balancing of the body, and also the thorax and abdominal segments, we can distinguish them separately because of the nature of its cuticle. There exists some difference with respect to the cuticle of the thorax segments and the abdominal segments. These are the peculiarities with regard to the general body pattern of the Drosophila, and we have seen the gastrulation process also. With this, I am going to wind up today's section. Thank you. I should fuck her in a Benz.